Welcome, friends. This is Mrinmay More, and I have chosen the topic Maxam Gilberts DNA sequencing. The first question we're asking in this video is what is DNA sequencing? DNA sequencing is the process of determining the nucleic acid sequence, that is, the order of nucleotides in DNA. It includes any method or technology that is used to determine the order of the four bases adenine, guanine, cytosine, and thymine. And the next question we're asking is who's the man behind it? Well, there are two men behind it, Alan Maxam and Walter Gilbert, who in 1977 to 1980 devised this method, and it was also named after them, Maxam Gilbert. This is also called as the chemical method because it involves the usage of many chemicals. The next question is there any other sequencing way for DNA? Yes, it is called Sanger dideoxy method after its inventor and also as chain termination method. Now let us differentiate between the two. Maxam Gilbert method is a chemical method, whereas Sanger is a chain termination method. Maxam Gilbert is highly sensitive and specific, whereas Sanger is less specific and sensitive. Maxam Gilbert uses base-specific cleavage of DNA by certain chemicals, 4 different chemicals, one for each nucleotide, whereas Sanger uses dideoxynucleotide triphosphates or DDNTPs as DNA chain terminators. Now let us see how this process goes through. It is also called as chemical degradation because it uses a lot of chemicals for degrading our DNA strand. The first step is denaturation, in which the double-stranded DNA is cut into single strands. Then comes radio labeling, and we label the 5' end of our strands with using a radioactive phosphate, that is P32. Then comes chemical degradation, in which the DNA strands are treated with various chemicals that cleave the specific nucleotides, helping us to decode the sequence. The next step involves agarose gel electrophoresis, in which the strands treated with varying cleavage chemicals are loaded onto high percentage page. Then next is reading the sequence, and that is which is very interesting. So let us get into the details. In this slide, we will understand the cleavage at specific nucleotides. The first reaction we will consider is the G reaction. The chemical used in this reaction helps to cleave the Gs. There are apparently 2 Gs. So that means 2 reactions will be required. The first reaction will delete this, second deleting this. Moving on to the next reaction is the A plus G reaction. In this reaction, both adenine and guanine are deleted. There is 1G, 1A, 2 reactions, 1G, 1A, 4 reactions. That means first we'll delete this, then this, then this, and this. Here we have 4 reactions. Going on, this is the T plus C reaction. That means this will be cleaving T and C both, thymine as well as cytosine. Here you can see there are 2 C's, 2 T's, 4, 5, 6, 7. That means we should have 7 reactions. Do we have them? 1, 2, 3, 4, 5, 6, 7. Yes, indeed, 7 reactions. Then the C reaction will cleave the C's. So, how many C's do we have? 1, 2, 3. That means we need 3 reactions to get rid of them. Here we have 1, 2, 3. So, 3 reactions. This is how the reactions take place, and then we load all of these chemicals into our gel. Moving on to the next step, fragments from the 4 reactions are arranged side by side in gel electrophoresis for size separation. Visualization is done by exposing to X-ray films for autoradiography. This gives us dark bands, each corresponding to radiolabeled DNA fragments from which the sequence can be inferred. Let us see the visualization that we get. Here is a chart which shows us the dark bands. On the left, you can see a size ladder which is in base pairs, 12 base pair, 11 base pairs. As you can see, the 12 base pair long strand is present in all of the reactions. This is because all of them have the first band. But further it varies. Now let us see how we decipher our sequence. In 11 base pairs, we can see visualization in T plus C and C. This means that our first nucleotide is common to T plus C and C. What is common between T plus C and C? That is cytosine C. That means our first nucleotide is C. Moving on to 10 base pair, again the similarity is between T plus C and C. That means our second is also C. Moving on to the 9 base pair, the visualization we get is for guanine G and A plus G. That means the common between G and A plus G is our third nucleotide, and that is G, as you can see. Moving forward, 8 base pair, the only visualization dark band that we get is for A plus G. The reason that it is unique for A plus G must be because it is not present in any other. This indeed means that there is something very unique to the A plus G reaction that we cannot find anywhere else, and that thing is A, adenine. That means our fourth nucleotide is A. Moving on to a 7 base pair, the only visualization we get is T plus C. That means there is something very specific in T plus C that is not present in any of the other reactions. What is very unique to T plus C? Yes, it is T, thymine. Therefore, we can see our next nucleotide here is thymine, and again after that we get visualization only in T plus C, and then again it is thymine and then C. This shows us how we decipher the code, and this goes on for another 2 or 3 rounds and we get our DNA sequence. This is how to read the sequence, and it is very interesting.